Hello, welcome back to MLT 110. In today's lecture, we're going to learn about renal physiology and urinalysis, and this corresponds to Chapter 5 in your intro textbook. Okay, we need to learn some different parts of the urinary system, and it contains these structures that are listed here on this slide. On the next slide, we'll actually see what these structures look like, but of course your urinary system is made up of two kidneys. These are the organs that form the urine. Next, below your kidneys, you have two funnel-shaped tubes that connect your kidney to your bladder. These are known as the ureters. The bladder works as a temporary storage location for urine before it's excreted. And finally, on this slide, we have the urethra. This is the canal that carries the urine from the bladder, so the urine can be excreted from the body. Okay, here as promised, here is the actual image of the urinary system. You have your two kidneys, you have your right kidney and your left kidney. Then below that you have the two funnel-shaped structures, the ureters that takes the urine from the kidneys down to the bladder, and then you have your bladder, which is like your holding tank for your urine, and then you have the urethra that helps carry the urine down to be excreted from the body. Also, something to note on this image is your renal vein and your renal artery. These are the structures that provide blood flow to your kidneys, blood flow to and away from your kidneys. On this slide, we're going to talk about components of the kidneys themselves. There's three major areas or regions of the kidney. First up is the cortex. This is the outermost layer of tissue lying just beneath the capsule of the kidney. The capsule would be just meaning a thin membrane on the outside of the kidney. We are going to be able to see a picture or a diagram of all these layers on the next slide. Next, lying just beneath the cortex would be the medulla. These are the portions or the layer of the kidney that contains the renal pyramids. These are triangular structures or cone-shaped structures that lie within this layer of the medulla. The third and final region or area of the kidney is the renal pelvis. You can kind of think of this as a funnel-shaped upper structure of the ureter. This is the place where the ureters start and where they attach. Okay, here is the diagram of the kidney. You have the cortex, which is again that outermost layer of the kidney. Next, just below the cortex, you have the medulla that contains those pyramid shapes or those cone shapes. Then finally, you have the renal pelvis, and the renal pelvis acts as a funnel. Everything flows to the renal pelvis and then eventually to the ureter. Also in this image, you'll notice you have your renal artery and renal vein pictured. And that's important because, again, you're going to have blood flow to and from your kidneys, because what are your kidneys ultimately doing? They're going to be filtering your blood. Okay, next up we're going to zoom in a little bit more on the kidney, and we're going to talk about some microscopic structures, some really tiny structures of the kidney. And with that, we need to talk about the nephron. The nephron is the actual functional unit of the kidney. Each kidney has approximately 1 million nephrons, which are located in the cortex. Remember, the cortex is that outermost layer. Okay, within the nephron, we have a few different structures that we need to be aware of. And the first one is the glomerulus. The glomerulus is a bundle of capsules. It's the basic filtering unit of the kidney. Remember, I just said earlier that we have blood flow to and from our kidneys, and that the function of the kidneys was to filter our blood. Well, this is where the actual filtration of the blood happens. It happens in these small little capillaries at a microscopic level that make up the glomerulus. Next is something called the Bowman's capsule, and the Bowman's capsule is a thin layer of cells that surround the bundle of capillaries that make up the glomerulus. Okay, next up is the tubules of the nephron. You have several tubules that are associated with the nephron. You have the proximal convoluted tubule, you have the distal convoluted tubule, and you have the loop of Henle. And each of these different tubules have a little bit different function, but mainly they're used for exchange of different elements. It might be water, it might be sodium, might be potassium, and we'll see what some of these tubules look like on the image that's coming up on the next slide. But each of these tubules are associated with each of the glomerulus, which will empty into eventually collecting ducts. And the collecting ducts will flow into the renal pelvis. Remember the funnel-shaped structure of the renal pelvis, which will then lead to the ureters. Okay, so here is your image of a nephron. Okay, remember we're zoomed in looking at microscopic components of the kidney. If you're looking here, you've got the kidney, and you can see like the cortex, and you can see the medulla and the renal pelvis. But if you just take this small little area and you magnify it, and here it is zoomed in and magnified, you can see you've got the nephron, and it's over here. And the first structure that we talked about was the glomerulus, and that was that ball of capillaries. It's the filtering unit of your kidney, the glomerulus. And what are we filtering in that glomerulus? We're filtering blood. Remember when you had that renal artery and that renal vein to supply blood in and out of the kidney? Next, you have the Bowman's capsule. Remember that thin layer right on the outside of the glomerulus, that thin layer of cells? Next, for our structures for the nephrons, we talked about the tubules. The first one coming out of the glomerulus was your proximal convoluted tubule. Proximal meaning closest to, and then you go down to your loop of Henle. Then you have your distal convoluted tubule, meaning distal meaning further away, and then they all go into the collecting duct. That was the last structure we talked about on the previous slide. The last thing I want to mention on this image is look at the different layers of kidney or regions of the kidney that the nephron is located in inside the kidney. This dividing line is in between the cortex and the medulla. Everything up here is located in the cortex. So the glomerulus and the Bowman's capsule and everything up here is located in the cortex of the kidney. But look down here, your loop of Henle is actually located in the medulla. And you can see over here on the image before it's really zoomed in, all this lighter colored image is in the cortex, and all this darker part of the triangle is in the medulla. Okay, so we've talked about the structure of the kidney and some functions of the kidney, but what's the actual process of urine formation? And there's basically 4 major steps to urine formation. The first step is filtration, and this step happens in the glomerulus and the Bowman's capsule. Things get removed like water, glucose, amino acids, and metabolic waste. The next step is reabsorption, and this happens mostly in the tubules where there's a transferring of water and other solutes through epithelial cells that line the tubules. Water and solutes are taken up by blood vessels that wrap around the tubules, and sodium absorption is a very important part of this step. The next step is secretion. So some molecules are actually released by the tubules, molecules like potassium and hydrogen. The last step is concentration. This happens to remove or to conserve water. And after the concentration step happens, the output for an average adult for urine is between 750 milliliters to 2,000 milliliters per day. Okay, now we need to discuss some different transport mechanisms for substances. We have something called active transport and we have something called passive transport. Active transport is where a substance requires energy or ATP to be reabsorbed. An example of a substance that requires energy or active transport is glucose. The process in which no energy is required for a substance to be reabsorbed is known as passive transport. An example of that would be water. Okay, we have some different terms related to urine composition and volume we need to discuss. I mentioned earlier the average daily output for a 24-hour period for urine would be about 750 milliliters to 2,000 milliliters for a person, but it could go as low as 600 milliliters on the low side. But a closer average without going to the far extreme on the low side or the high side would be 1,200 milliliters to 1,500 milliliters per day for a person. The first term we're going to discuss is polyuria, and this is when a person has excess production of urine, so it would be in excess of that 2,000 milliliters per day. Next is oliguriria, and this would be a decreased production of urine, so it would be less than 600 milliliters per day. Next is anuria. This would be an absence of urine production and could indicate failure of kidney function. Nocturia is the last one, and this is an excess urination at night, so if you have to get up and go a lot and pee during the night. The next concept we need to discuss is renal threshold. This is when a substance is not normally excreted by the kidneys, but it ends up appearing in the urine because the blood concentration is above a level that the body can hold. An example of the renal threshold would be when the body's blood glucose level exceeds the range of 160 to 180 milligrams per deciliter, and the glucose would be excreted out in the urine because the body is holding as much as it possibly can, and that glucose beyond 160 to 180 has to go somewhere, so it comes out in the urine. So that's known as the renal threshold. So we as lab techs can collect several different types of urine specimens for testing. The first type of testing is just the random urine sample. This test can be collected, or sample can be collected any time of day. It's used for just routine testing, and to detect obvious abnormalities in the urine. Next is the fasting or first of the morning collection. The first of the morning specimens tend to be more concentrated, so this is the sample that's usually more preferred. This is usually when the pH of the urine is a little bit more acidic, and it will help preserve any cells that may be present within the urine. Next is the clean catch or midstream clean catch urine sample. These are usually used for urine cultures when you're trying to eliminate contamination in your samples. You're usually given a towelette to clean before you're to collect the urine sample. Also, you may be asked to eliminate a little urine before the sample is collected. All this is done to try to help eliminate contamination in your urine sample. Next up are your timed urine samples, and the most common one would be a 24-hour urine sample. This would be where you would collect all your urine within a 24-hour period, and of course you would have to be sent home with a urine container that would be able to handle the volume of a complete 24-hour sample. The last type of urine sample we need to discuss is the catheter urine sample, and these are usually used for culture, and these are usually collected when patients are unable to urinate on their own, and the catheter sample is really good to eliminate contamination. There's some criteria that we want to follow when we're doing specimen collections of urine samples. For our urine samples, we want to use a disposable, wide-mouth, flat-bottom container, and we want to use a container, if possible, that have screw caps. We want to use a container that are clear, and containers that can hold at least 50 milliliters in capacity. For pediatric samples for babies, we may need adhesive bags in order to adhere it to the baby's bottom. For 24-hour urine specimens, we would need larger plastic containers in order to hold the volume. And then finally, we would need to wear gloves when we're working with urines because we have to follow universal precautions and consider that everything could possibly be infectious. After we have collected our urine samples, we have to be worried about storage and preservation of our samples. We usually want to test our samples within 2 hours of collection. If for some reason we're not able to test within 2 hours of collection, we need to find some way to preserve the urine sample before testing. An ideal preservative would be something that is a bacterial side that would prevent bacterial growth, and it inhibits urease formation and preserves formed elements within our urine sample. The most routine method of storage and preservation for our urine samples is just refrigeration, and this is a must for our samples that we're going to culture. But some things to take into consideration when you do refrigerate your urine samples, that there could be a precipitate of amorphous crystals that form while they're in the refrigeration. And before you test your urine samples, if they have been refrigerated, you should let them come back up to room temperature before you perform any chemical testing on your sample. There are some commercial transport tubes available for urine samples. You just have to make sure that the tests that you're going to be testing your urine sample for are compatible with the preservatives and things that are in those tubes. This is the end of part 1 of the urinalysis lecture. I hope you learned a lot about renal anatomy and a little bit about urine samples and urine collection. I will see you over in part 2.